Hello and welcome back. Today is part one of our exploration into genetic technology. This field could be a class in itself. It is so broad. There's so much going on in it. So we are really just scratching the surface here, but we're going to focus on two main forms of genetic technology today. And then in part two, we'll do kind of like a brief overview of a number of other big types of genetic technology, big things that are happening in the genetic tech world. So let's just make sure we're all on the same page here. What is biotechnology? So that just refers to using living systems like DNA, right? Something that is alive to develop technology or to make a product. Okay, so sometimes this is called genetic engineering. We're going to really refer to it as biotech though, but just like if you see this in passing, biotech is kind of interchangeable with genetic engineering. So there's tons of different types of biotech. I kind of talked about that already. You can see on this image that there are these are just like broad categories of different types of biotechnology. So like you can go into the health field, food, aquaculture, which is like dealing with oceans, deserts, agriculture. This can also be used for like biowarfare. There's just like, at this point, this is like the big field right now. Everyone's doing this. The science is getting crazy. We're learning a lot of what we as a species can do, and so this field is sort of like exploding in popularity right now, which is really cool and exciting. And so let's get into a few of the big types of biotech. I guess before we actually do that, we need to learn a little bit more about like vocabulary here. So one big thing that you're going to hear over and over again is recombinant DNA. And this is how you get one species DNA into a second species DNA. And when you do that, it's called recombinant DNA. So this is the joining together of DNA from two different sources. So you get a piece of DNA that is of interest. Maybe it's something for a plant that is like resistant to pests or something. And then you insert it into a vector, right? This could be like a virus, this could be like a bacteria cell, just something that will replicate quickly. And then you get this recombinant DNA. So now this is the gene of interest in this vector that's going to replicate very quickly. And then you can insert this new recombinant DNA into whatever species you want, and then it will, take it in, that new species, and use whatever this gene of interest is. It's really, really interesting, but recombinant DNA is how you get the gene of interest into a new species, right? You can't just like take DNA from like one thing and just like insert it willy-nilly, right? There has to be a process, and this is step one of the process, is making recombinant DNA to then put into the species that you want to change. And when you do that, when you receive genes from another species, that species is now called transgenic, okay, because it has a different set of genes. So transgenic, different set of genes. So genes from one species are inserted into another to make fast synthesizing proteins. So DNA produces proteins, right? It goes through a few steps, which we'll talk about in the upcoming video notes. So DNA goes through a few steps and produces proteins, and those proteins are what make you, right? It gives you all of your general characteristics. The example that is on the screen, though, is insulin protein for diabetes patients. So in humans, you have a gene that produces insulin. And insulin is how you can maintain your blood sugar. It's how you can break down sugars when you have too much, or get sugars back into your blood when you're feeling a little tired. Okay, so not all humans can, go through this, create this insulin gene, right? People that have diabetes don't have this or a working insulin gene. So we can take this gene from a human cell and place it into a bacterial plasmid. And look, now we have a recombinant plasmid, right? We took the gene of interest and we put it into this vector, right? This plasmid, this is vector, okay? And then we put it back into the bacteria. You can kind of see down here, it's got that insulin gene, right, at the bottom here. So now this bacteria is transgenic because it has genes from 2 different species in it, right? A human gene and then its normal bacterial gene. We're gonna grow this gene, we're gonna grow this bacteria, excuse me, in culture because bacteria replicates like every 20 minutes. It's so fast. So we're gonna put it in culture and we're gonna replicate it tons of times where we have just like unlimited now at this point insulin, and then we can extract that insulin, and that's what people use if they can't make their own insulin. Like if they have diabetes, this is how they can get insulin, by going through this process. Okay, so it's really interesting, and this is one great example of how we get or use biotechnology. Now, some other big uses of genetic engineering is genetically modified foods, or GM foods. You may have heard of these before, GMOs, in passing on the news. There are some risks and benefits to these. We're gonna go way more in detail, so I'm not gonna spend a ton of time on this slide, but just so that you're aware, some risks are introducing allergens or toxins or accidental contamination between genetically modified and non-genetically modified foods. You can create antibiotic resistance. You can change the nutrient content of a crop in either a positive or a negative way, and so that could be a risk. And then a creation of super weeds, which are like really hard to kill weeds, and then environmental risks, right? But some benefits are increased pest and disease resistant, which allows your plants to grow more. They can be drought tolerant, so they can grow in more arid climates, which means that they could maybe grow longer or in different areas where they typically can't grow. And this would lead to an increased food supply worldwide, which is a huge pro, in my opinion. Another type of genetic engineering that we're going to focus on in this set of notes is gene therapy and genetic screening. So gene therapy is this idea of fixing or adding mutated DNA or proteins. You can do this to cure genetic diseases by replacing the defective or missing gene by adding in a healthy gene. So if you have a cell with a defective gene, you can introduce the healthy gene instead, and then you would have the cell function restored because now your cell is going to, base its decisions off this healthy gene rather than this defective gene. So that's gene therapy, is fixing or adding in a healthy gene. And then genetic screening is kind of what it sounds like. You are screening your genes to determine if you have genetic diseases. This is really good to know if you are ever planning on having children. You can know the risk for passing on disease, or if you just want to know about yourself and to see if you have any risk of disease. So that's genetic screening. Now, the issue here, and this is a big hot button issue, is the ethics of genetic engineering. And what are ethics is kind of like morality. Do you think something is morally right or wrong? Do you know the consequences of your actions? Do you think that this is the right choice, this is fair, this is honorable, right? At this point, science is moving fast, and we don't always know the final outcome of science. So this conversation of ethics is an ongoing conversation, and it's important to take part of this conversation because we don't always know the consequences of our actions. And so we need to make a decision personally, is this morally right or wrong? And so some of the big questions of questioning the ethics of genetic engineering is producing harmful organisms, right? They can be accidentally produced. Is that a risk we're willing to take by doing all of these experimental science experiments? Is producing harmful organisms, is that okay? Like, yeah, we're gonna do a lot of really great things, but we might accidentally do something harmful. Is that okay? I don't know. It's an ethical issue that we need to discuss. Increasing the ability to diagnose genetic diseases before birth. Okay, that's another kind of like ethical, legal, social consideration, right? Because this can add a lot of complexity to the abortion controversy, right? If you know going in that your child is going to have a lot of genetic diseases, those are decisions that you and your partner need to be talking about and making, and that is a major ethical issue. This is something, again, you have to decide. The last ones that we're going to talk about, just like again, I want you thinking about this. I want you to really be conscious of these topics as we talk about them, because you're really going to have to focus in on these ethical issues on our next project. Should we modify genes in humans? Is that okay? At what point is, like, are we going too far here? Like, looking at this cartoon here, like, do you want to choose your child's talent? Like, oh, I want to have, a musical genius for a child. Yeah, I could modify genes to do that. Is that okay? I don't know. Is it fair if genetic screening and gene therapy are very expensive? They are wicked expensive. So that maybe puts a limit on who can and who cannot access these different genetic engineering tools, right? So if, Jeff Bezos, I don't know, can afford all of this, but, someone else can't, is that fair? Again, I don't know. I want you just thinking about this. And then the last major ethical issue is, how can we control whether people make biological weapons using biotechnology? I don't have answers to any of these questions, but they're really good thought topics, and they're really good things to be discussing with your classmates, discussing with yourself, and making decisions on where you lie on these issues. So great job. I know this was a little bit of a long set of notes, but I think it's really important and it's really interesting. So I can't wait to dive further into this in class. I'll see you then.